Good morning. Welcome to my channel. Today I'm going to discuss about glycogen metabolism. In the glycogen metabolism, we can see the glycogenesis and glycogenolysis, two cycles. Before going to glycogen metabolism, first I'm giving little information about glycogen. What is glycogen? Glycogen is an animal storage homopolysaccharide consisting of only alpha D glucose. Glycogen is made by only alpha D glucose. Glycogen is mainly stored in liver and muscles of animals. It is highly branched structure. It also has alpha 1,4 glycosidic linkage, alpha 1,4 glycosidic linkage in the straight chain, and alpha 1,6 glycosidic linkage at the branching point. So the glycogen has both the linkages, that is alpha 1,6 linkage in the straight chain, alpha 1,4 linkage at the straight chain, alpha 1,6 linkage at the branching point. In between any branching points there are about 12 to 18 glucose residues, we can see it. And innermost core of glycogen contains a primer protein, or also called as glycogenin. This glycogenin is a innermost core of glycogen. Okay, it is also called as first molecule of glycogen. Okay, this glycogenin is also called as glycogen primer. So as I told, glycogen is stored in liver and also in the muscles. The liver glycogen is converted into glucose during the fasting and gives energy to the body. Then this muscles glycogen is converted to lactic acid during the exercise and gives energy to the body. These are the little information about glycogen. Next we'll come to metabolism of glycogen. So as I told, in the glycogen metabolism we are studying two cycles. One is glycogenesis and glycogenolysis. So glycogen is an animal storage homopolysaccharide. It is synthesized during feed condition and breakdown during the fasting condition. So it is synthesized during the feed condition, that is called as glycogenesis. Formation of glycogen from glucose, it's called as glycogenesis. Breakdown of glycogen to form a glucose, it is called as glycogenolysis. Okay, so this glycogen is stored in cytoplasm of liver and muscles. Around 100 grams of glycogen is stored in liver and 250 grams of glycogen is stored in muscles. At the time only one cycle will occur, that is either glycogenesis or glycogenolysis will occur. So in this video I am discussing only glycogenesis. So glycogenolysis I am discussing in the next class. So what is glycogenesis? Definition: formation of glycogen from glucose, it's called as glycogenesis. So this cycles mainly occur in liver and muscles. The intracellular site is cytoplasm. Okay, so the starting compound is glucose and the end product is glycogen. So next we'll come to reaction pathway. In the reaction pathway there are three steps. One is activation of glucose, glycogen synthase, branching enzyme. These are the three steps we can see in the formation of glycogen cycle. So first we'll study about activation of glucose. Glucose is a starting compound. Glucose is converting into glucose 6 phosphate in the presence of glucokinase in liver, hexokinase in muscles. Here ATP is converting into ADP. This ATP donates phosphate molecules to glucose. The glucose will become glucose 6 phosphate. So ATP is converting into ADP, adenine diphosphate. This reaction is based on phosphorylation and it requires magnesium as a cofactor. And Next is glucose 6 phosphate is converting into glucose 1 phosphate in the presence of phosphoglucomutase. This reaction is based on isomerism. Next is glucose 1 phosphate is converting into UDP glucose in the presence of UDP glucose phosphorylase. So this UTP, uridine triphosphates, releases 1 phosphate molecules, and glucose 1 phosphate releases its phosphate molecules. So 2 phosphate molecules are releasing. So this glucose joining with UDP, UTP is converting into UDP because it releases 1 phosphate molecules. It will become UDP. UDP is joining with glucose molecules, so it will become UDP glucose in the presence of UDP glucose phosphorylase. This UDP glucose is a donor of glycogen synthesis. The second step is glycogen synthesis. This glycogen synthesis is one enzyme. By the action of this glycogen synthesis enzyme, UDP glucose donates its glucose molecules to glycogen primer to form glycogen, and free UDPs are liberated. Okay, this UDP is it takes 1 phosphate molecules from ATP and it will become UTP. That UTP is joining with glucose 1 phosphate and 2 phosphate molecules are releases. Again it will become UDP glucose. Okay, so the third step of activated glucose will repeats again and again and forms UDP glucose. This UDP glucose donates the glucose molecules to glycogen primer to form a glycogen. Okay. Activated glucose units are sequentially added by the enzyme glycogen synthase. The glucose units is added to non-reducing end of glycogen primer to form an alpha 1,4 glycosidic linkage, and free UDPs are liberated. Next step is branching enzyme. This is also one enzyme. Okay, when the chain has been lengthened to minimum of 11 glucose residues, a second enzyme, the branching enzyme, transfers a part of alpha 1,4 chain to neighbor chain, producing a new alpha 1,6 linkage, those forming a new branching point. Then the branches grows by further addition of glucose in alpha 1,4 direction by glycogen synthase enzyme, and then further branching by branching enzyme. See here, the UDP glucose donates the glucose molecules continuously, and the chain of the length will become around 11 to 18 molecules. When the length of the chain will become around 11 to 18 molecules, the branching enzyme transfers the part or around 6 glucose residues to next chain or neighbor chain. Okay, and this branching enzyme forms one linkage, that is alpha 1,6 glycosidic linkage. This branching enzyme creates the branching point. That branching point is alpha 1,6 glycosidic linkage, and again by the enzyme glycogen synthase enzyme, the length of the chain will grow. The glycogen synthase enzyme helps to add the glucose molecules. The length of the chain will grow, and again the branching enzyme will react. It will transfer the part of the chain to next chain or same chain. So again many branches will occur by the repeating action of glycogen synthase and branching enzyme. So finally many branches will occur, that is called as glycogen. Here the green color molecule is there, that is glycogenin. It is a one innermost protein. It is also called as first molecule of glycogen. And here in the glycogen synthase, Two important enzymes are glycogen synthase and branching enzyme. Glycogen synthase enzyme can add glucose units only in alpha 1,4 linkage, and this branching enzyme is needed to create the alpha 1,6 linkage. Okay, this branching enzyme is required to create the linkage. The glycogen synthase enzyme is required to add the glucose molecules in alpha 1,4 glycosidic linkage. This is about glycogen synthase cycle. Next we'll come to significance of glycogen metabolism. The liver glycogen is required to maintain the blood glucose level in between meals, and muscles glycogen is used exclusively by muscles during the contraction. All the enzymes related to glycogen metabolism are cytoplasmic. It means the cycle will occur in the cytoplasm. Thank you for watching my video. Please like, share, subscribe my channel. Thank you.